We've lost the media and we've lost the adventitia or the adventitious sparse, and basically what we have is an endothelial tube, and that endothelial tube is very thin and allows us to get nutrients across it or waste products across it. When we talked about epithelial cells earlier in the quarter or even last quarter, we talked about epithelial cells always have a basement membrane. So if we looked at cuboidal epithelial cells, we always have a basement membrane that anchors it and holds it together. There's glycoproteins and stuff, a few other stuff. And so when we get into the capillaries, that starts to even thin out. And we basically just have, these are columnar cells. We're talking about squamous cells when we're getting into the capillary, when we're talking about the blood vessels. But we still start to lose that basement membrane because it gets in the way of diffusion, both across, both ways, in and out. And then some of our blood vessels are going to be, the majority of them are continuous, meaning the capillaries, the cells are held together by tight junctions, kind of like we saw in the heart. So these continuous ones are going to be abundant, going out through the skin, muscles, everywhere, when we're spreading blood out to the tissues. They are leaky, meaning that stuff can get across, like nutrients and oxygen, and we can get fluid back into them and fluid out of them. So all the nutrients and stuff that we want to exchange with the muscles and the skin and the other tissues get out because capillaries are very thin. But in some places where we need to get larger proteins in, hormones in, nutrients in or out, we're going to poke holes in these capillaries, and those are the next ones that are fenestrated. Fenestrated is, I believe, from the Latin word, in French we'll call a window, fenetra. Drapes, when you're doing surgery, they'll call them fenestrated drapes. They have a hole in them. And so basically we have these holes in this capillaries. And so we do see this basement membrane. It's thinner than it usually is. It may be breaking up. We have our tight junctions are holding all these cells together so they don't tear apart, but they're very fragile, so we have to have very low blood pressure when we're in the capillaries. And then this is a continuous one, so we don't see too much, but if we look at the fenestrated ones, they're going to basically be where we need to have larger, more permeability, because larger things like proteins or hormones are going to be getting in or out of the blood supply, and they can't squeeze between the tight junctions, so we have these holes in them, windows in them, fenetra, so they're fenestrated. That's going to allow lots of hormones or proteins or bigger molecules to get in easier, in and out. But that's still not good enough when we're trying to cleanse the blood. So this is a relatively large capillary because you can see the size of these red blood cells. Sometimes the capillaries are even smaller. These red blood cells are giving nutrients to the tissues, and some of the nutrients in the plasma, in this case, need to get through larger openings. But if we want to clean this, let's say we want to destroy that red blood cell. We talked about red blood cells dying when they get old. They go through the liver and the spleen, and the liver and the spleen have to pick those red blood cells out and destroy them and digest them and reprocess them. And so we can't really get into this through these kind of capillaries. So we open up even more and make big gaps inside between the cells, and those are our sinusoids. So if you look at a sinusoid, we have these big gaps where if we have a macrophage sitting right here, it can grab this red blood cell and devour it. Or if there's toxins or waste products in here. So anytime we're going through a system like the bone marrow where we want cells to get in and out of the bloodstream, it's got to be open like this. Or in the liver where we're going to take out lots of bad stuff like dying cells or toxins. It's like almost, I exaggerated even more, it's almost like a trough. These blood vessels are almost open enough that they're going through a trough, and they're lined by usually macrophages that are picking clean like a factory line. And you're taking out things that are bad. So the production line's moving along, and you're standing there, and it's like, oh, this chocolate's bad, I'll take that one. I always think of Lucille Ball. So think of the general concept. You don't have to memorize where we're going to see all these things, but obviously wherever we need more permeability to get stuff into or out of the blood, we're going to have a sinusoids. And if it's just a little more permeable than a capillary, we're going to have fenestrated ones. But for the most part, they're pretty tight or continuous. Because we don't want proteins getting out of the blood, because remember, when proteins get out of the blood, water follows and we can get edema. So if we're taking water to the capillaries of the skin and proteins getting out, or even red blood cells are getting out, we can get swelling. We don't want that. So we got to keep a lot of the stuff in. We do want things like oxygen or amino acids to get out. So we do have a thin membrane, but we don't want everything getting out, unless it's in a place where we can handle it, like the liver or the spleen. So a lot of times when we look at blood vessels, we see vascular shunts. The classic one from the artery to the vein is called a metarterial thoroughfare. That's a big term, but it's what it's called, I call it as an anastomosis. And anastomosis is where one vessel joins another. And we talked about that when we talked about the heart, so that if one vessel gets plugged up in the heart, it doesn't block blood flow to that area. We can get blood through another vessel coming back to the same area. And so anastomosis will be where we connect blood vessels to blood vessels. So the metarterial thoroughfare, it's a big term, but like a thoroughfare, I guess, on the highway goes straight from one city to the other. We can go right straight through from the blood side, the arterial side, to the venous side without really any change in the blood. It'll just go straight through there, and that would be our thoroughfare. If blood's coming through this vessel and this blood vessel's plugged, blood can come around this way and get to the tissues over there, so those tissues don't die. Or if this is blocked, or if this is blocked, let's say you got a clot blocking this whole area, we can still drain these tissues because they can work their way around it through these anastomoses. So the anastomosis gives us alternative routes for blood to flow. This also shows us the arterial. So arterioles do have a lot of that media or muscle layer that can control how much blood flow is coming into this capillary bed. So if we change the blood flow, we constrict it down and very little blood flow comes in here. Very little blood's getting to this tissue bed. So we can cut off blood flow to the next capillary bed. Everything downstream from this arterial can be cut off by changing the diameter. Or if this tissue is really active, like it's thigh muscle and you're doing squats, we open this blood vessel up, and now blood comes flying into these capillaries, and we're bringing lots of nutrients, lots of oxygen, and that's increasing blood flow, or what we call perfusion, to the tissues. All that blood flow can come in, and well, we just saw these capillaries are one cell thick. They're very delicate. Their basement membrane's not really that strong. They don't have an adventitia. And if we have all that blood flow coming in here, it could rupture these capillaries. So we have these precapillary sphincters that can control the pressure inside the capillaries by constricting. If too much blood's coming in, they can pinch down and they can shunt some of the blood through or to the other side. And so there's these little independent valves or gauges on each little branch of the capillaries that can determine how much blood flow is coming through this capillary. So we can control how much blood flow is coming into the main tissue, like the thigh, through these arterioles. We can even do it higher up on the distributing arteries. So some of the arteries that are bigger can shunt blood away from this leg entirely. And we do that when we're digesting. We're going to shunt it away from the legs and the muscles, send it to the gut, or the opposite if we're running. We send it away from the blood, from the gut, and we send it to the muscles. So those are the bigger distributing arteries. But when we get down to the thigh muscle, let's say we want to send blood flow to the rectus femoris and maybe not the hamstrings as much, then these guys come into play, and they can open up and say, all right, he's doing squats, we need to send more blood flow to the front half of his leg or whatever. But on an individual fiber basis, if we're looking at the rectus femoris and it's got a lot of blood flow going through, we can send blood flow to different parts of that muscle belly. Some are going to be maybe more active than other fibers, or other fibers might need more oxygen or more waste products. Or if there's an issue, we can change the flow by changing the precapillary sphincters. So these are changing blood flow to the tissues, the arterial are changing blood flow to the tissues, and those distributing arteries are changing blood flow to the tissues. So we can change blood flow to tissues in 3 different ways. Here we see that when we cut those precapillary sphincters off, now blood's not going to go through this bed. It's just going to pass right on through to the venous side and then be carried away from that tissue altogether. And we do that sometimes if this tissue's bogged down, can't handle any more nutrients, oxygen, or blood flow, or the opposite. If this needs more blood flow, they can relax and we can let more blood into that area. So depending on the needs. Those needs are going to be later in the chapter, but if you think about it, what would increase blood flow? Why would we want to increase blood flow to this tissue area? So metabolically, if the oxygen level starts to drop, we're going to relax these sphincters. We're going to open this up and bring more blood flow in because low oxygen, hey, the tissues are screaming for oxygen. Or increased pH, I should say a decreased pH, increased acidity. So if you're generating lactic acid because your muscles are running, the pH drops, these guys can change the amount of flow into the tissue. And so if the tissue demands go up for nutrients, we vasodilate and increase blood flow. We control this stuff based off of the pressure. We control this one by neurological mechanisms. Blood flow through the capillaries. So the precapillary sphincters regulates blood flow into the capillaries. Blood flow is regulated by vasomotor nerves and local chemical conditions. So you can either bypass or flood the capillary bed depending on your needs. So if for some reason the oxygen levels are dropping, we can vasodilate and bring nutrients in. We can relax the capillary bed. The capillary bed is going to be based right there locally off of what's going on in the tissues. But vasomotor means neural control. So the nervous system gets involved and says, hey, we need to shunt blood away from this entire leg. We got to send it to the gut because the person just ate and we need to digest food. When we get across the capillary bed, we start to collect the blood that we've been giving the nutrients away. So we've given off oxygen. We have deoxygenated hemoglobin. We have carbaminohemoglobin now. We've picked up some other waste products, and we have to take it back to the lungs to blow off the CO2 or the liver to process it. We collect them in little venules, or tiny veins, little veins. And then the venules start forming venules start forming our veins, and then eventually either end up in the superior or the inferior vena cava, which are our largest veins. As we move away from the capillaries, we're going back to the heart. And what we see here, we have a blue heart today. When we're looking at blood pressure coming out of the heart in the lab, we did this. I said the person we talked about was 140/90. That means we have 144 millimeters of mercury pushing blood out to the capillaries. When we get down to the capillaries, we have about 40 millimeters of mercury. The reason blood leaves the heart and goes to the tissues is a difference in these pressures. 140 millimeters of mercury here and only 40 there. We have 100 millimeters of mercury difference. That's the driving force that pumps blood to the capillaries. When we get to the capillaries, it's moving fairly slow. There's a lot of friction, and we drop another 20 points or so, and we get down to like 20 millimeters of mercury when we cross the capillary bed. So in other words, the beginning of the veins, the pressure inside our veins is now only 20 millimeters of mercury. That's a low pressure vessel. We were up at 140. Some people have higher blood pressure than that. But 90, 100, 120, that's a lot of pressure compared to just 20 millimeters of mercury. We're over here now. We want to get back up to the heart. When we dump into the heart, right before we go to the heart, we're at 2 to 0 millimeters of mercury, meaning no pressure at all in the right atrium. So the only force that's driving blood away from the capillaries and back to the heart is 20 millimeters of mercury. And that's not enough pressure to push blood back to the heart. So one of the things that we see in the vein side is that if we get blood to move this way, we don't want it going back down. If it's talking about, if this is a muscle in my calf, and the blood has to go from my calf and it's trying to come back up to my heart, it's going against gravity. That's not enough. 20 millimeters mercury can't push blood back towards my heart. And so If we do get it to move up towards the heart, we don't want it to come back down, so we have valves in veins that we don't have in arteries. And these valves open and I'll let blood go through, and then when blood tries to come back down through them, the valves close off. We have two other ways of helping blood return to the heart. One is whenever you contract your muscles, they squeeze these tubes, and it's like a tube of toothpaste. You squeeze them down, and we force blood up. So if you have a muscle belly, and a muscle belly, and a vein running up through it, when you contract your muscles, they squeeze this vessel and they force blood back towards the heart. Then they relax. We don't want the blood to come back where it went because we have these valves. So we have a muscle pump. That helps us get blood from our feet up to our head. Don't have to worry about it when it's coming from your head down to your heart, unless you're standing on your head. If these valves get leaky or stretched out because of water volume problems like pregnancy or high blood pressure or poor vein health, these valves stretch out and then the vessels get really, really wide and big, and that's varicose veins. It happens in your legs. It's hemorrhoids if it happens in your mud. Not too much fun. So the muscle pump. The other way that we get blood to come back to the heart is the respiratory pump. The respiratory pump is breathing. When you inhale, you're decreasing the pressure in your lungs, and the pressure in the atmosphere is greater, and air is actually forced into your lungs. You're not sucking air in, you're creating a negative pressure by stretching your rib cage out. It's the same thing as stretching an accordion out. When you stretch an accordion out, air gets sucked into the accordion. And when you squeeze down on it, it forces the air out. So when you stretch the thoracic cage out, it sucks air into your lungs. And then when you squeeze your thoracic cage down, usually your diaphragm contracts, but if you blow hard, you contract your thoracic cavity and force the air out. But when you're inhaling, because you're creating a negative pressure and it's pulling air in, it's also sucking blood in from the vena cava. So by decreasing the pressure in your chest, by just doing that, not only did I just suck blood in, air into my lungs, I sucked blood into my heart, and that's the respiratory pump. So if you're running on a treadmill and you're getting tired, if you breathe harder like that, you're actually assisting. You have the muscle pump from your legs contracting, but you're assisting blood flow by the respiratory pump. You can see that in people who are foolish enough to get married, when they're standing up in front of the minister, and their muscles are rigid, so they're not squeezing them, they're not moving, they're scared to death because they're making the biggest mistake of their life. And their heart is pumping 100 miles an hour, so it's pumping all the blood out. How's it getting back up to their heart? Gravity can't do it, they're not moving, and they're barely breathing. It doesn't, so it pulls in their feet, doesn't go to their brain, they fall over, they can't say I do, they win. Yep. You shouldn't be doing this. You also see it in people in the military a lot of times, or PE. They have you run, 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 run. Your heart's beating, all this blood's going out, your muscles are contracting, and you're breathing hard, and it's pumping it all back to your brain, and everything's all good. And then they say, line up and attention, and they go like this, and they hold still, and they're like, what's happening? Their heart's still beating fast from the exercise. All the blood's being pumped out, but they're not contracting their muscles, and they're not breathing like crazy, so they can't get the blood back into the heart, and then they pass out too. And so, gotta keep your moving. That's why when I walk up here, I move all the time. I squeeze my blood vessels, it pumps blood to my brain. My brain's happy. My brain thinks it's happy.